Hi, friends. Good morning. We are back for our next section of Module 9 Evolution, part scientific theory, part unconfirmed hypothesis. And so here we are on page 285. We are at the section titled Molecular Biology, the Nail in Macroevolution's Coffin. What a title. Are you ready to go? Here we are. I'm ready. All right. Of all the scientific data that provide evidence against evolution, perhaps the most important come from the field of molecular biology, which studies the properties and structures of the molecules important to biology. Think back to what you learned in the 3 previous modules. Aside from DNA, what is the most important type of molecule in the chemistry of life? The protein. As a result, a large amount of the research effort in molecular biology centers on understanding proteins. Early on, molecular biologists noticed something rather amazing. There are certain proteins that are common to many species. Most animals, for example, have the protein hemoglobin. This protein transports oxygen through the bloodstream to the cells. In addition, most organisms have the protein cytochrome C, which takes part in cellular metabolism. Interestingly enough, these proteins are not identical from species to species. In other words, the cytochrome C that you find in a bacterium is a bit different from the cytochrome C that you find in a human. How do these proteins vary from species to species? Well, what determines the structure and function of a protein? In module number 5, you learn that the sequence of amino acids within a protein determines its structure and function. Thus, if you were to examine the amino acids in the cytochrome C of different species, you would find slightly different sequences. For example, Table 9.1 lists the amino acid sequences on the same portion of cytochrome C for several different types of organisms. There's the horse, the kangaroo, the human, the chicken, the tuna, the moth, and yeast. All different. Now, remember what we are looking at. Remember what we're looking at in this table. Each 3-letter abbreviation represents a specific amino acid. Thus, gly stands for the amino acid glycine. Lu stands for the amino acid leucine, and so on. Don't worry about the NH2. That's an amine functional group that really has no bearing on our discussion. Also, notice that some of the 3-letter abbreviations are red. Red amino acids indicate that they're different from the amino acid expected if you use the horse sequence as the standard. Thus, compared to the horse sequence, the kangaroo sequence has 1 amino acid that is different. However, the yeast sequence has 4 amino acids different from the horse sequence. What do we see in studying Table 9.1? Well, first notice that all of these sequences are very similar. That's not at all surprising because the protein is the same in each case, cytochrome C. It performs the same basic function in each organism, but in order to be able to work with the specific chemistry of each organism, it is slightly different in each specific case. That's where the differences in the sequences come from. The cytochrome C section, shown here, for example, is nearly identical between the horse and the kangaroo. The only difference is the second amino acid, which is leucine, leu, in the horse, and isoleucine, ile, in the kangaroo. Because of this one difference, the cytochrome C of a horse will not work in a kangaroo or vice versa. What does all this tell us? Well, think about how proteins are made. They're made in the cells according to the instructions of DNA. Thus, by looking at the amino acid sequences in a protein that is common among many species, you're actually looking at the differences between specific parts of those organisms' genetic code, the part that determines the makeup of that protein. If macroevolution is true, then that portion of the genetic code should reflect how closely related the two species are. If the two species are closely related, the DNA sequences that code for a common protein should be very similar. If they're only distantly related, however, the DNA sequences that code for that same protein should have more significant differences between them. Looking at the differences between the amino acid sequences of a common protein, then, is a way to determine just how many differences exist between corresponding sections of the DNA of the organisms in question. For example, the portion of the amino acid sequence for cytochrome C shown in the table is 11 amino acids long. Of those amino acids, there's only one difference between the horse and the kangaroo. We can therefore calculate the percentage difference between the cytochrome C amino acid sequence in a horse and the cytochrome C amino acid sequence in a kangaroo. Percent difference is 1/11 times 100 equals 9.1%. Comparing the amino acid sequences in cytochrome C for the yeast and the horse, however, there are 4 differences. Thus, the percent difference is 4/11 times 100 equals 36.4%. These two comparisons then tell us that the portion of a horse's genetic code that determines the makeup of this part of cytochrome C protein is much closer to the kangaroo's than it is to a yeast's. From a macroevolutionary point of view, this would tell us that the horse is more closely related to the kangaroo than to the yeast. In other words, some macroevolutionary scheme in which one life form evolves into another, the kangaroo would be closer to the horse than would be the yeast. Well, that makes sense, doesn't it? After all, a yeast is considered by macroevolutionists to be a rather simple life form, whereas kangaroos and horses are rather complex. As a result, it makes sense that a horse is more closely related to a kangaroo than a yeast. Even though comparing this portion of cytochrome C in the horse, kangaroo, and yeast makes sense in terms of macroevolution, as you look across the data that is out there, you find that it causes serious problems for macroevolution. Consider, for example, the bacterium Rhodospirillum rubrum. Rhodospirillum rubrum. When its cytochrome C amino acid sequence is compared to vastly different organisms, nothing makes sense in terms of the macroevolutionary hypothesis. Table 9.2 shows the percentage difference between the amino acid sequence in a Rhodospirillum rubrum cytochrome C and the amino acid sequence of other organisms' cytochrome C. Now, remember what macroevolution says. It says that complex life forms evolved from simple ones. Well, the simplest life form on the planet is a bacterium. Of the organisms listed in the table, the yeast, a single-celled fungus, is probably the next simplest life form. Increasing in complexity, then, come the silkworm moth, followed by the tuna, followed by the pigeon, followed by the horse. Thus, macroevolution would assume that the bacterium is most closely related to the yeast, then to the silkworm moth, etc., etc., all the way up to the horse. As a result, then, the yeast cytochrome C should be most similar to that of the bacterium. The silkworm moth cytochrome C should be the next most similar, and so on. According to the data, however, each organism in the table is essentially as closely related to the bacterium as any other organism on the table. If anything, the bacterium is more closely related to the most complex organisms, not the least complex ones. In other words, the data presented in Table 9.2 show none of the evolutionary relationships that should exist if macroevolution really occurred. Instead, these data seem to indicate that the bacterium is just as different from the horse as it is from the yeast. As you look at more and more data like this, you will find that this is the pattern of the vast majority of the data. Regardless of the protein studied, the amino acid sequences seem to indicate that each individual type of organism is just as different from one type of organism as it is from another. Just to make it clear that the data is really overwhelming on this point, we present a few more tables like Table 9.2. Now, think about what these data tell us. We are comparing the cytochrome Cs of a mammal, horse, a bird, pigeon, a reptile, turtle, a fish, carp, and an eel, lamprey. According to macroevolution, eels would have come first. Later, fishes would have evolved, followed by amphibians, followed by reptiles, followed by birds, and finally followed by horses. Thus, the eel should be most similar to the fish, then the reptile, and then the bird, and it should have the least in common with the horse. Instead, the data tell us that the cytochrome C in the lamprey eel is most like the carp, but then next most like the horse and has essentially the same differences between the turtle and the pigeon. If you look at the data presented in tables 9.2 and 9.3, it is clear that you can establish no macroevolutionary trends. This is the case with the vast majority of the data collected from molecular biology. If you map the amino acids of virtually any protein and compare the differences between organisms that have that protein, you will generally find no macroevolutionary trends. Instead, Each kind of organism seems to be equally or nearly equally different from every other kind of organism. As is the case with all of science, there are exceptions to this general rule, but those exceptions are quite rare. Even though the exceptions are rare, some macroevolutionists actually highlight those exceptions as evidence for evolution. You see, if you pick and choose your data very carefully, you can find examples of molecular biological data that seem to indicate a macroevolutionary trend. For example, look at Table 9.4. The rhesus monkey, the rabbit, the horse, the tuna, the screwworm, the wheat, the yeast, and the bacterium. Table 9.4 is titled A Highly Selective View of Some Cytochrome C Sequences Compared to Human Cytochrome C Sequences. Now, this table does seem to indicate a macroevolutionary trend, doesn't it? After all, as you go down the table, you are getting to simpler and simpler life forms. As the life forms get simpler, their cytochrome C sequences seem to get more and more different from that of a human, don't they? The problem with this table is that in order to construct it, you must ignore 99% of the data from molecular biology and choose only the 1% of the data that agrees with the hypothesis of macroevolution. This is clearly not responsible science. In science, we must look at all of the data, and the data from amino acid sequencing provides strong evidence against macroevolution. All right, guys, great reading today. I'll see you next time in the middle of page 289 with the subject heading Macroevolution Today. Happy day to you. Bye.